Hello everyone, we are the R&D team of Genosensor. I am Dr. Deepika and I am working as research head at Genosensor, and these two are my colleagues of R&D team.
Hello, my name is Satpreet Kaur and I am working as a research assistant at Genosensor.
Hello, I am Ashika Verma and I am working as a research assistant at Genosensor.
So this is our sepsis panel kit in which there are different cocktails and the probes for infection and antimicrobial detection. So this is the sepsis panel kit. These are the cocktails that we will use for sample processing, and this is probe box that has been designed for infection and antimicrobial resistance as per the specific species or gene. And this is the kit booklet, and it is recommended to read this kit booklet thoroughly before starting the experiment because every necessary information has been mentioned here. So this is the kit booklet that has been provided. Also this is the cassette which will be provided with this kit. So in single cassette we can keep 12 sensors. So at a time 12 samples can be analyzed, one sample on each sensor. So this is our PRATHAMASENSE Genosensor IVD kit that has been designed for the sepsis panel along with the cassette docs. So we have done pre-preparation for the experiment. Before starting the experiment, it is recommended that it should be processed in the dark. So we will switch off the light of the cabinet, of our biosafety cabinet. So as per the instructions, we will process sensors as well as the samples simultaneously. So this is our urine sample. So we will process our sensors and the samples simultaneously. So these are the sensors. As per the protocol, we will broadcast cocktail 1 on this sensor. Now we will dispense cocktail 1 onto the sensor. We will dispense this sample onto the circular area only, which is our working area, and you should be careful that you don't touch the tip with the sensor area. So you have to be careful. After putting cocktail 1, we will keep them undisturbed for next 10 minutes, and now we will process our sample. So this is our urine sample. What we will do, we will take 1 ml of the sample in the Appin drop and will centrifuge the sample. So this is our next step. That's what I was telling, that we have to process sample as well as the sensor simultaneously. Now we have taken 1 ml sample. We will send these samples for the centrifuge for 10 minutes.
Now we will centrifuge the sample to pellet down for 10 minutes at 14,000 rpm.
Now after 10 minutes, we will add capture probe to the sensor surface. So these are our probes. Now we will dispense the probe onto the sample. We have to be very careful. There should be no frothing, there should be no touching of the sensor area. So we will dispense the probes 10 microliter and we will mix thoroughly 10 to 20 times. There should be no bubble, no frothing. Now we got the samples after centrifugation. It's been now 10 minutes. What we will do, we will discard the supernatant and will process the pellets with cocktail 2A and cocktail 2B as per the instructions. Now we have added cocktail 2A and cocktail 2B to the pellets and we have mixed the sample thoroughly 10 to 20 times. You must take the precaution that there should be no frothing during the mixing. Now this sample will again go for the centrifugation for next 15 minutes.
Now we will centrifuge these samples for 15 minutes at 14,000 rpm to ensure the cell lysis.
So after 30 minutes we will wash the surface of the sensor with cocktail 3 like this. We have to take precaution so that we don't touch the working area of the sensor, which is the circular one. Now we will keep the sensor aside. Now we got our samples after centrifugation. In this step we will collect the supernatant, and in the previous step we have taken the pellet, but here we will take the supernatant into the another APN drops, and in these supernatant we will add our detector probes. Now after adding the detector probe to the supernatant, we will keep this sample in the incubator at 65 degrees centigrade for the 10 minutes. Now we will keep these samples in the incubator for 10 minutes at 65 degree temperature. Now we have taken out the samples after 10 minutes from the incubator. These samples will be drop casted onto our sensor surface for the second hybridization. In this sample we have added detector probe, and here on the sensor surface we have added the capture probe. Now this sample will be drop casted onto this sensor surface, so there will be hybridization between the capture and the detector probe. So I will add 10 microliter of this sample onto the sensor. Now we will keep this cassette into our humidifier box, and we'll keep that humidifier box into the incubator. So this is our humidifier chamber. This is the plastic box that can sustain the temperature up to 65 degrees centigrade, and these are the ice packs. So this is our humidifier chamber. Like this we will keep this whole box in the incubator. The temperature is same 65 degree, but this time it will go for 15 minutes. So after 15 minutes we have taken out this cassette and our sensors. We'll add next cocktail onto the sensor, which is cocktail 4. So we will add cocktail 4 onto our sensor surface, which is a circular one. After adding cocktail 4, we will keep it undisturbed for the next 10 minutes. After 10 minutes, we will wash the sensor again with the cocktail 3. Washing is done with cocktail 3. So we will wash the surface areas of the sensor like this. You don't have to touch the surface of this sensor, so you have to do washing very carefully. Now our 90-minute protocol is over. We have processed our sample as well as we have processed our sensor. Now this sensor is ready to be analyzed with the PRATHAMASENSE Genosensor device. So this is our processed sensor that we will analyze on the PRATHAMASENSE Genosensor device. I have inserted the sensor in the slot very carefully. Now I will add the developing solution onto the sensor surface. Make sure that developing solution get dispersed over the whole surface, working reference counter. So you have to disperse the developing solution over all the 3 electrodes. Now we will analyze this on the PRATHAMASENSE APK. This is our single channel APK, as we are analyzing the single sample. So this is our added patient. We will go to generate report, and regarding the panel, we have different panels. In this case, as we told earlier, that we have processed as per the sepsis panel, and we have processed the samples for antimicrobial resistance. That's why I have taken fungus none, and for bacterial infection again I will select none, and in case of antimicrobial resistance I have selected NDM, because we have processed the sample for NDM detection and the probes has been used towards NDM detection. Now I will go for connect device and will so here we will go to high peak and then peak data. So this data we can export in the Excel, so we can save our raw data. From here we will generate the report that will be saved in the PDF format. So we have saved the report. Report has been downloaded successfully. So this, you can see the report at the top, PRATHAMASENSE sepsis panel, and regarding antimicrobial resistance, NDM is found to be positive. NDM has been detected as per the current values, and in the result analysis also antimicrobial resistance NDM detected. So this is the format of report.